for biology classes. I am S.K. Roy here. So we will continue with photosynthesis. Some question we'll solve. Okay. So here is one question that is the carbon dioxide to NADPH2 ATP ratio in C3 plants. What is the ratio of the carbon dioxide to NADPH2 to ATP in C3 plant? All right. So here is there a different ratio has been given. This how many carbon dioxide for the reduction of the carbon dioxide, how many NADPH2 molecules are required and how many ATP molecules are required. Actually, in the dark reaction of normal plants in Kelvin cycle, there is for one molecule of carbon dioxide to reduced into sugar, into sugar, it requires this here 2 ATP, 2 NADPH2, and one molecule of ATP also is required for the regeneration of RUBP. This is a regeneration. Okay. Regeneration, and this one is the reduction process. All right. So for the reduction per carbon dioxide molecule, 2 ATP, 2 NADPH2 and another ATP. So it is one carbon dioxide molecule require 3 ATP molecule and 2 NADPH2, and that's why the ratio is carbon dioxide to NADPH2 ATP. The ratio, our answer will be A, that is 1:2:3. That for one carbon dioxide, 2 Oh, okay. So we'll continue. There was a problem due to internet. So for one molecule of carbon dioxide, 2 NADPH2 and 3 ATP are required per molecule. And as for one molecule of hexose sugar, there require 6 molecule of carbon dioxide. So this 6 molecule of carbon dioxide to reduce, then 6 to the 12 NADPH2 and 18 ATP molecules are required. So for this production of one molecule of hexose sugar, that is glucose or fructose, it's require 6 carbon dioxide 2 NADPH2 and 18 ATP. Ratio remain the same. If you find out the ratio is 1 is to 2 is to 3. So our answer is this one. This thing in case of C4 plant, say this is C3 cycle. This one is C3 cycle, all right, and C3 plant. So here this in C3 plant requirement is less, but as in the C4 plant, there is additional ATPs are required. In this case, this ATP molecule requirement is more. So here 1 carbon dioxide, 2 uh, 2 NADPH2, H2 to 5 ATP molecule. All right. This is for C4 plant. C4 plant has higher requirement of ATP. Okay. We'll go to the next question. Next question is the similar question in C3 cycle for the fixation of every carbon dioxide molecule, the reduction and regeneration step required. Here is the answer: 3 ATP and 2 NADPH2. Okay, same question that we have already discussed. This is you can this is abbreviated forms of the that C3 cycle. Okay, the C3 cycle, and here this one C3 cycle, or this is also known as a Kelvin cycle. Okay, Kelvin cycle, here is carbon dioxide is absorbed, and that is the carboxylation process. Okay, ribulose 1,5 bisphosphate that absorb, and it produce 3-phosphoglyceric acid, and this 3-phosphoglyceric acid is reduced to this one triose phosphate. This compound is actually PGAL, that phosphoglyceraldehyde. Okay, and from here the sucrose or a starch is produced. Here the ATP and NADPH is utilized, and then this process is the regeneration. During the regeneration also ATP is utilized. This is the same thing that I have drawn here. Okay, the same thing I have drawn: carbon dioxide and Rubisco enzyme. Not this. This is the Rubisco enzyme has taken place for every time. For the RUBP is the carbon dioxide acceptor, and first stable compound is the 3-phosphoglyceric acid. This is a 3-carbon compound, and that is why this cycle, Kelvin cycle, is known as C3 cycle. Okay. Next question, there is one: when Rubisco act as an oxygenase, what will happen? See, this is one of the important thing with the C4 plant. in C3 plant that they which carry out the normal photosynthesis, that Rubisco is the enzyme. This Rubisco enzyme sometimes function as oxygenase, and also it function as carboxylase. Mainly it function as carboxylase, but sometimes it function as a oxygenase, means instead of absorption of the carbon dioxide, it help to combine with the oxygen. So when oxygen is absorbed instead of carbon dioxide, then there is a process that phosphoglycolate glycerate and phosphoglycolate are produced. Phosphoglycolates are produced, and in this case what happened that during this process, when it function as a oxygenase, there is a process which take part. That process is known as photorespiration. When it function as oxygenase, it is the initiation of photorespiration, and in the photorespiration, phosphoglyceric acid and phosphoglycolate or the phosphoglycolic acid is produced. This phosphoglycolic acid is the initiation of photorespiration, and when this photorespiration takes place, the net productivity of the photosynthesis is reduced. That is a less amount of mm, sugar or carbohydrate is produced. So this is for photorespiration takes place in C3 plant, and this is due to Rubisco enzyme. The Rubisco has the dual nature, that it can function as oxygenase, it can function as carboxylase. Next question. Next question is the 22 number. The reaction of Kelvin cycle do not directly depend on the light, but they usually do not occur in night. Okay, this is this is Kelvin cycle. This is the actually the dark reaction. Is it? This is a dark reaction because the process of carbon dioxide reduction is independent of light. It does not require light, but still this process of dark reaction or the Kelvin cycle, it does not take place at night. It takes place only during the daytime. So what is the cause that it is? The cause of this is this one. This assurance that Kelvin cycle is dependent on the product of light reaction. All right, this is, it depend on the product of light reaction, and that's why the dark reaction, though it is independent of light, it takes place during the daytime, not at night, because it require this for the reduction of the carbon dioxide, it requires ATP and NADPH2. it requires NADPH2, and these two are the product of the light reaction. So at night, these products are not available for the carbon dioxide reduction. So the Kelvin cycle also takes place during the daytime. Abar gelo bodhoy. Hello, student, can you hear me? Hello?
Yes, sir, we can hear you.
Yeah, okay. I thought the internet problem is there. Question number 3. Which one of the following statement is incorrect for carboxylating enzyme in C3 plant? Okay, carboxylating enzyme in C3 plant. The carboxylating enzyme means that is the Rubisco. All right. Or we can call it RUBP carboxylase. Carboxylase oxygenase. Okay, this so with this oxygenase also has been added due to its dual nature. Okay, so this is carboxylating enzyme in C3 plant. Which of the following statement is incorrect about this carboxylating enzyme? Say this one, bifunctional nature. Naturally, this is correct. Rubisco function as a carboxylase as well as oxygenase. Can bind only with carbon dioxide? No, this one is the wrong statement, because this Rubisco, it can bind with carbon dioxide, it can bind with oxygen. So this statement, the wrong statement about the Rubisco is can bind only with carbon dioxide. This is the wrong statement. Otherwise, its old name was carboxymutase. It's located in the stroma of the chloroplast. Naturally, this RUBP carboxylase or Rubisco, this occur in the stroma of the chloroplast, because stroma of the chloroplast, because the Kelvin cycle, site of Kelvin cycle is the stroma of the chloroplast. So this is correct, this is correct statement, this is correct statement. This statement is wrong, that it can bind only with carbon dioxide. Next question, very easy one, that ATP as well as NADPH2. Okay, both are required during the conversion of, I think you remember that this NADPH2, ATP and NADPH2. You should remember this one, that Kelvin cycle, this ATP and NADPH2 are required during the conversion of phosphoglyceric acid to the phosphoglyceraldehyde. Okay, so this is the reduction step, and so our answer will be this one, that PGA to PGAL. Next question, we will move quickly. Which of the following statement is wrong with respect to C4 plant? C4 plant, I think you remember that some plant this which one is wrong regarding the C4 plant? Plants that are adapted to dry tropical region have C4 pathway. They tolerate high temperature and show a response to high light intensities. Their leaves show crans anatomy and they undergo photorespiration. Say this one is wrong statement. Here actually wrong statement we have to find out. So this statement is wrong, and this one is correct. They are adapted to usually all the C4 plants. They are in the dry areas and where the high temperature is there. So they are from the tropical. They are usually tropical plants. C4 plants are tropical plant. Okay, but remember that all the tropical plants are not C4 plant, but C4 plants are tropical plants. Okay, so they tolerate high temperature. They have greater productivity of biomass. Okay, this is also correct, very correct. Their productivity, they can produce more carbohydrate, they can reduce more carbon dioxide to carbohydrate. They are more in this way, they are more adapted. They at very low carbon dioxide concentration also. This C4 plant can produce carbohydrate through the hatch and slack cycle. They carry out C4 cycle, and this way their efficiency, productivity is very high. This statement that see their leaf shows cranch anatomy, this is true. In C4 plant, we find the cranch anatomy, a special type of anatomy in the leaves. But they undergo photorespiration, this is wrong. So in C3 plant, in C4 plant, photorespiration is nil. Photorespiration is almost nil. Okay, because they have one carboxylating enzyme which do not combine with the oxygen, they combine with carbon dioxide only. So this is our answer. Come to next question. In C4 plant, the in mesophyll cell, C3 acid is transported from bundle cell. Say, for this, in C4 plant, actually there is two step. In C4 plant, there are two carboxylation process. Carboxylation reaction. One takes place initial carboxylation that takes place in the mesophyll cell. And the second carboxylation, that is the final or the second all right, and here is the final reduction of carbon dioxide. Of carbon dioxide takes place, and here in the in C4 plant, Kelvin cycle occur in the bundle sheath cell, all right, not in the mesophyll cell. In case of C3 plant, the light reaction in C3 plant light reaction and dark reaction both the mesophyll cell, okay, but in case of C4 plant, the light reaction in mesophyll light reaction dark reaction which is by the Kelvin cycle takes place in the bundle sheath cell. Okay, so here in case of C4 plant, the light and dark reaction has been separated in two different types of cells. That is in the mesophyll cells chloroplast. Okay, this there is this bundle sheath cell. I think you remember that in the leaf there is a vascular bundle. Okay, so vascular bundle, the veins are there, and around the vein there are, say, around the sheath cells. So here this in the bundles itself, C4, this Kelvin cycle operate. So carbon dioxide reduction in the bundles itself. C3 acid, so sugar is formed in Kelvin cycle occur in bundles itself. So our answer is this one. In C4 plants, Kelvin cycle occur in bundle sheath cell. Next one, decrease the rate of photosynthesis at high concentration oxygen is referred to as, this one is known as the Warburg. This is known as Warburg effect. That is, at a high concentration of oxygen, the rate of photosynthesis is reduced. That Warburg was a physiologist. He reported first these things, that effect of oxygen on the rate of photosynthesis. 28. Particularly large cell around the vascular bundle of C4 plants are called, that just now we have learned that it is bundle sheath cell. Next question, in C4 plant, carbon dioxide combined with PEP in presence of, say in case of C4, in case of C4 plants, okay, there are two carboxylation that we have learned, and the initial carboxylation is carried out by the enzyme called PEP carboxylase, which take part this PEP carboxylase in presence of carbon dioxide plus water is in presence of PEP. PEP is phosphoenol pyruvate. This is phosphoenol pyruvate. This PEP, in presence of PEP carboxylase, combine with carbon dioxide and produce oxaloacetic acid. Okay, this oxaloacetic acid is a C4 compound, that is 4 carbon compound. This contain also 2 carboxylic group, so dicarboxylic acid also it is known as, and then this oxaloacetic acid is converted into malic acid. This malic acid is transported to the bundle sheath cell, and there, by the breakdown of the malic acid, this carbon dioxide is released, and that carbon dioxide is absorbed in the Kelvin cycle to reduce it to carbohydrate. So C4 plant, carbon dioxide combined with PEP in presence of next question. Number 30, in C4 plant, Kelvin cycle occur in where? Bundle sheath cell, stroma of the bundle sheath cell in C4 plant. Okay. Next is Next is tropical plants like sugarcane shows a high efficiency of carbon dioxide fixation. High efficiency sugarcane. Okay. Sugarcane, maize, panicum, panicetum. Okay. The some member of Amaranthacea. These are the some of the important C4 plants. All these are tropical. Most of the C4 plants are in Gramineae, in the family Gramineae. Okay, that modern name is Poaceae. Gramineae, that where in the under the Gramineae family, all our that food grain plants belongs. Okay, all this rice, paddy, wheat, maize, this belongs to Gramineae. But these C4 plants, most of the plants, C4 plants are Gramineae, but it is not that only the monocots have the C4 plant. Some dicot plants also have this C4 cycle. So tropical plants like sugarcane with high efficiency of carbon dioxide fixation because of Hatch and Slack cycle. All right, this is Hatch and Slack cycle is also known as C4 cycle. Okay, this Hatch and Slack cycle, but due to so it is C4 cycle is Hatch and Slack cycle. Say here, this one is say this is the diagrammatic representation of this uh, C4 plants and C4 cycle. Say here that this atmospheric carbon dioxide is atmospheric carbon dioxide is uh, carbon dioxide is absorbed in presence of phosphoenol pyruvic acid that fix it, and it forms a C4 C4 acid that I told oxaloacetic acid. Oxaloacetic acid is converted into malic acid. Then this malic acid is transported to the bundle sheath cell. This bundle sheath cells are not that much colored. Okay, chlorophyll is very less in this. That's why it has been shown non-green almost. And here it this C4 acid is broken down. It releases the carbon dioxide. This carbon dioxide is absorbed in the C3 in the Kelvin cycle, and then this sugar starch is synthesized. After that, this C3 acid, which is produced after releasing the carbon dioxide, this is again transported back to the mesophyll cell, where it is again changed into the phospho enol pyruvate. This one absorb the carbon dioxide from the environment and then it transport it to the bundles itself in the form of acid. Okay, this cyclic pathway is known as Hatch and Slack cycle. And that is it. Okay, all right, Hatch and Slack cycle. In C4 plant, in which cell or tissue we can find the starch more? Okay, where will we find a starch? Whether it will be mesophyll cell or bundle cell or epidermal cell. See, a starch is produced, carbon dioxide is first reduced to sugar, that is and ultimately it is changed into a starch. A starch is the reserve food for the in in the C4 plant we will find the starch more in the bundles itself. Okay, not in the mesophyll cell, but in case of C3 plant, in C3 plant it is mesophyll cells will have more accumulation of the starch, but C4 it is the bundle cell. Next question, bundle cells have all of the following characteristics except which one is not the characteristics of bundle cell? They have bundle cells are generally large size. This is true. Okay. Large number of chloroplast. No. Thick wall impervious to gaseous exchange. This is this is also gaseous exchange they don't require. They have a large number of chloroplast, of course, for the stroma is required, but they don't have much grana in the chloroplast with intercellular space. Bundle cells, they don't have intercellular space. Okay, this one. Next is this is primary acceptor is in the mesophyll cell. All right. For photosynthesis in CAM plant. Cam is another plant which shows a special adaptation for high temperature. This cam plant crassulacean okay, acid metabolism. This is another kind of the plant which grow in the desert areas and dry areas. Dry regions. All right, these are the succulent plant. Succulent plant like bryophyllum and other plant where the leaves they have thick leaves and fleshy leaves are thick and fleshy. This is shows another type of adaptation. In these also more or less C4 like cycle operate, but these plants for the uh, high temperature adaptation they do not open their stomata. They open their stomata open at night. Okay, but for photosynthesis it requires the daylight. So what they do at night, they open their stomata, absorb carbon dioxide, change it into organic acid and store it throughout the night, and during the day when there is a light, this organic acid break, that malic acid, oxaloacetic acid, they break into organic acid and source of carbon dioxide for photosynthesis in CAM plant also is the malic acid. Remember that in the C4 plant also malic acid. Initially, oxaloacetic acid is formed. Oxaloacetic acid is converted into malic acid, and it is in case of CAM plant, this malic acid is stored throughout the night, and in the during the daytime malic acid break and there is a question related to the Kelvin cycle again. Why Kelvin cycle is called C3 cycle? Okay, this is because that the first stable compound is a C3 compound. This is a PGA, phosphoglyceric acid. So this is the first stable compound is a 3-carbon compound, and so this one is known as C3 cycle. Kelvin cycle is known as C3 cycle. Okay, next question. The PEPKs. PEPKs means PEP carboxylase. Okay, PEPKs in brief, it is PEP carboxylase, which is found in C4 plant and also in CAM plant. All right. This is not PEP carboxylase is absent in C3 plant. Rubisco is found in all the plant, of course, in C3, C4, and CAN, because this is the enzyme of the Kelvin cycle. Okay, and Kelvin cycle is the main Kelvin cycle. Kelvin cycle is the main process of the reduction of carbon dioxide. PPKs has the advantage over the carboxylase is that PP carboxylase function only as carboxylase, never as oxygenase. So PPKs is PPKs and with oxygen, but PPKs does not. See, Rubisco, this one will be the answer. This that Rubisco combined with oxygen. Rubisco combined with carbon dioxide as well as oxygen, but PPKs never combined with oxygen. They combine with only with the carbon dioxide. So this is the advantage, and due to this, that the C4 plant has in the temperate region, more humid condition, temperate region with more dry condition. Sorry, not the temperate, it is tropical region. This will be the answer. The tropical region with more dry condition. C4 plants are C4 plants are common in the tropical dry condition. Next, 39. C4 plants are able to minimize photorespiration because this we have already learned that C4 plant in C4 plant photorespiration is almost nil, and photorespiration takes place in C3 plants. But why the C4 plants have minimized the photorespiration? Because in this C4 plant there is a additional cycle, that Hack and Slack cycle, and in that the PP carboxylase or PPKs initiate carbon dioxide fixation. C4 plant. This is one question, very important question, and here you see, read the following statement. Here some 5 statements are given, and all these are the characteristics of the plant, certain group of plant. They have a special type of leaf anatomy. They tolerate higher temperature. They shows a response to high light intensities. They lack photorespiration. They have greater productivity of biomass. Okay, all these all C4 plants, they are they have a special anatomy. The anatomy is known as Cranch anatomy. What is Cranch anatomy? They have Cranch anatomy. Cranch anatomy, that is the bundle sheath cell, the ring of bundle sheath cells. This is special in the C4 plant. Ring of bundle sheath cell and then uh, in their chloroplast, grana is less and stroma is more. Chloroplast less grana. Why less grana? Because this chloroplast bundle cell do not carry out light reaction. Light absorption is not required, so those grana organization is also not required. They carry out only Kelvin cycle dark reaction, so the stroma is there. Okay, they tolerate higher temperature. Yes, this is they tolerate high temperature adapted to. They show response to high light intensity also. At higher light intensity and high temperature, they can carry out their photosynthesis efficiently, and they lack photorespiration. This is very big advantage, and they have greater productivity of biomass. All these characters we find in the C4 plants. Okay, so this is an important question showing this one is matching, that correct matching, which one is correct for C4 plant. In C4 plant, see, PPKs is there which carry out the C4 cycle, and Rubisco is also there in the bundle cell which carry out C3, which carry out the C3 cycle. Okay, so Rubisco for the C3 cycle and PPKs for the C4 cycle in the mesophyll cell. So our answer is this one. That is the answer. Next is number 42. Okay, the photosynthesis of maize plant differ from wheat plant in. So the question here is difference between the photosynthesis of maize plant and wheat plant. What is the difference? Here the question has been asked in a different way. Actually, this maize plant is, that we have already learned that maize plant is a C4 plant. All right, and the wheat plant, this one is C3 plant. All right, so you can say that the difference between the photosynthesis of C3 and C4 in another way, but here the you are tested for that whether you know which one is C3 and which one is C4. So additional knowledge is first thing. If you don't know that this maize is a C4 plant and wheat is a C3 plant, then rest of the portion you cannot do. You cannot proceed. So this is you must know. Photosynthesis of maize plant differ. Number of ATP required per carbon dioxide fixation. Say in C4 plant, ATP requirement is more. Do you remember that there is in C3, it is 1:2:3, but in C4 it is 1:2:5, 5 that ATP. Is it? This carbon dioxide, NADPH2 and ATP. CO2, NADP, NADPH2. Please read it as NADPH2, and this is ATP. So here the ratio is a requirement of ATP is 3. per carbon dioxide. Here it is requirement is more. So here altogether for one molecule of glucose or fructose to synthesize, it requires 30 ATP molecule. Having a spatial separation in both carboxylation. This is also true. Okay, this number of ATP requirement is high in maize plant. There is a spatial means there is a separation by space. Then having PEP carboxylase activity. Maize plant has PEP carboxylase activity. This class, you can, this video will be available in the YouTube. So later on, you can ask the question. Okay. Thank you.